Walking down the street, we get the funniest looks from everyone we meet.
Hey, hey, we're the monkeys.
Hey, people say we monkey around, but we're too busy saying hey to put anybody down.
We go wherever we want to, do what we like to do. We don't have time to get restless, there's always something new. Hey, hey, we're the monkeys.
Hey, people say we monkey around.
Hi everybody, this is Dr. Ed Park from Recharge Biomedical Clinic, and today we're doing a webinar on Parkinson's disease. The symptoms of Parkinson's are a tremor, which is kind of a rhythmic shaking of the extremities. This comes from a loss of inhibition. There's a circuitry in your brain which inhibits movement, and in the absence of it, there is just baseline movement. The human frequency is somewhere between 4 and 6 cycles per second. In addition to tremor, there's slowness of movements. You can't really do stuff you want to do precisely and quickly, and that's because of a loss of the direct pathway. Direct pathway is activating. Third cardinal sign is postural instability. You can't balance, and that's because standing and balancing is a delicate operation. It's literally a balance of awareness in space, of muscle tone, of movement, and so because the interaction between suppression and activation is bad, the circuitry is bad, and you can't maintain posture well. There's also the fourth sign is rigidity, meaning the arms usually and all the other extremities are rigid. If you try and move them passively, there's resistance, even like cogwheel resistance. And as the disease progresses, there is depression, and it can even lead to dementia, although that could be just from the brain getting older. The posture is bad, often because of osteoporosis, but also because they have to look down and get information about where their feet are, because the whole feedback loop between what we call proprioception, meaning position in space, the brain and everything is bad. And some people find it actually easier, instead of trusting their internal sense of movement, awareness, posture, to actually look at as if the feet were somebody else's feet, and that helps them walk more steadily. Here is the tremors. You can also get like a pill rolling movement, like you're rolling a pill between your index finger, your fingers and your thumb, and you shuffle. The thing we need to realize about Parkinson's disease is that it really isn't a disease. It's just a syndrome. When I think of a disease, you think of something like polio, or in the case of, let's say, Lou Gehrig's disease or amyloidotropic sclerosis, it's a genetic disorder which causes a specific thing. Parkinson's, on the other hand, can be caused by toxins, pesticides, heavy metals, trauma such as boxing, with, in the case of Muhammad Ali and other greats, stroke, and again, there's no single genetic cause. In fact, I think as we get towards this, I'll argue, as I do in all of these lectures, this being the 23rd, that aging in most disease is a case of replicative senescence, which means that as the cells copy and copy and copy, they acquire errors in aging, and when that occurs, the cells and then the organs fail. There was a gentleman, he wrote a famous treatise on neurological injury and regeneration. His name was Ramon y Cajal, and he did not say this, but people interpret him as saying that neurons don't really regrow well. And we'll challenge that as we go on, because it's key to understanding how Parkinson's develops and how it can be cured, per se. This is a clutch. A clutch is something that couples a moving part with another part, and it needs to regulate the way they interact. So in a way, the damaged pathways in Parkinson's disease are a damaged clutch. And those are the neurons in the substantia nigra, or the black substance, which we'll just briefly touch upon. This cartoon illustrates that it says, your car will be ready in a moment. The mechanics is saying the last rites now. And what that's meant to suggest is that cars are like people. The parts will fail, and if we don't refurbish the parts or replace them, we're in trouble. People think of disease as something that is a mandate from God or a genetic lottery. Not so. You wouldn't go to your auto dealer and say, well, I do have a family history of muffler problems, or, I drove it 1,000,000 miles, but I didn't expect anything to go wrong. This is just accumulated wear and tear. One concept that I came up with, I haven't heard a lot of people talk about, but it seems to enter my mind every time I prepare one of these webinars, is agency. So agency is an idea of where does the problem occur? Where's the problem? So again, we look at Marvin the Martian looking down on Earth, and he might see people, let's say, doing an election in an election year. And he'll say, oh, the problem is the Republicans or the Democrats or democracy or a constitutional republic or whatever. He doesn't know where the level of importance is. He might see these things. Oh, it's the food, it's the money, it's the ideas, it's the ability to get to the polls that's important. But really what he should be looking at is the people. People, Republicans, Democrats, Libertarians, they're going to the poll. They use the ideas of money, energy, all these things to get to the polls, and they create things like democracy or republicanism, the Democratic Party. But the level of agency is the people. Similarly, with all these cell diseases, and most diseases are cell diseases, the true level of agency is on the cell level, those being neurons and glial cells, which are supportive players to the neurons. So damage and dysfunction is always it's written into the DNA and to the code, which is the epigenetics. And we did a thing on epigenetics with identical twins in podcast 21 that you can check out. So most of the literature, most of people's understanding is looking at the tools of these cells or people in this analogy. They think of neurotransmitters like dopamine. They think of inflammation. Maybe there's a viral cause or exuberant inflammation. They look at the pathological accumulation of these Lewy bodies, and they try and tweak the pathways to fix and understand and make models of Parkinson's. But I don't think that's where the action is. These are tools used by the cells. And similarly, they look at things like Alzheimer's and Parkinson's and senility. Again, disease being just an emergent property of cellular dysfunction. If you get enough cells bad in your coronary arteries, you get a heart attack. The heart attack doesn't cause the bad cells. So let's dial back and look at what Marvin might misinterpret as the problem. Well, look, we have a failure of these substantia nigra, these cells in these pathways in the basal ganglia. So basically, dopamine is lacking. So let's push a bunch of levodopa, which is what people have taken for many years, and L-dopa is converted into dopamine, so you just push in more substrate into the reaction, and the remaining cells that are there get better fuel, and they reverse or they help correct the symptoms of Parkinson's. But that's like taking an 8-cylinder car, trashing 6 cylinders, and then throwing rocket fuel or 100 octane into the remaining 2 cylinders. Better to have 8 cylinders. 2 other things that patient will see later is taking is selegiline, which is a monoamine oxidase inhibitor. MAO is the way that these catecholamines are degraded. So if you get rid of the degradation, or if you block up the drain, then the bathtub gets more filled with neurotransmitters. Mirapex is a dopamine, DA, an agonist, and again, this somehow helps, although the whole idea of receptor saturation, regulation, drug holiday is an issue. And we know that if you keep on taking levodopa, it's orally administered for years, then it stops working in a thing that some people call tachyphylaxis. It just, the receptors get numb to it, so you need a little holiday for them to reset, because they just get like a callus on the skin. Other effective treatments, although not without risk, are deep brain stimulation, which is like a pacemaker for the brain. They implant a device up there, and it sends signals that somehow distract or modulate the problem. And then stem cell transplants, which have had great promise in experimental and animal models, because Embryonic stem cells, although controversial, retain an ability to differentiate into ways that we need them. So they have replacing the broken or dead cells. So what is a neuron? A neuron is a cell that is differentiated to send signals. Okay, so here on the dendrites, these are little fern arms. They have a bunch of little listening devices, and a listening device is a synapse. A synapse is a gap. Okay, so you get the receiving end here. I'm listening for all my signalers. The nucleus is of course the information of the DNA coding for this one single cell. You got the axon, which is the cabling running down here. You got a little bit of coding, which is the insulation around the cabling, and then you have the outgoing signal. So the axon ends at these little terminals, and they dump neurotransmitters such as dopamine or what have you into this gap. It's kind of like a kneecap, you know, the end of your tibia. It sits in a floating space, the synovial joint. Same thing, it dumps transmitters. The transmitters are received here, and the signal is transmitted electrically. So electricity, neurotransmitter chemical release, neurotransmitter docking, more electricity. Okay, so let's look at cells. Cells are stem cells, which we talk about in podcast 5. These are self-renewing progenitor cells. It's kind of a black box, but suffice to say that even though the neurons, which are the stars of the show, are what we think about in terms of degenerative neurological disease, the oligodendrocytes and the astrocytes are also the nursemaids. These are like the roadies or the crew that help set up the show. So they're all important to function. And the interesting thing is they all come from the same progenitor. So we've always understood that these things are reproducing. That's where most of the brain cancers come from, the nurse or the helper cells. But similarly, the neurons regenerate. So that whole thing of Ramon and Cajal and the thing that they indoctrinate into both medical students and frat party goers is that neurons go and they don't come back. Well, that's not true. So nerves do regenerate. In fact, the process of dreaming every night, you form about 10,000 new brain cells, and that's because the memories, the feelings, the emotions of the previous days and years, every time you go to sleep, it's recoding and you're figuring out what's going on, and that requires hard coding. And so forget the idea that you don't regenerate nerves, because you do. All right, this slide, forgive the visual pun, but in a nutshell, is all you need to know about Parkinson's. Truly, the subject of neuroanatomy is infinitely complex, and it's not really that important to understand for our purposes. What we have is an area on the outside surface of the brain that moves stuff, and there's this area adjacent to it that feels stuff. So if you open up a head case, a cranium, and touch it with little probes, someone will feel their hand, or you can touch the probe and their leg will move, and it will always be the same. They're mapped out in these sectors. Well, this part feeds down to the deeper brain called the basal ganglia, and this is where the information is coordinated. Okay? In this particular structure, this kind of Brazil nut-shaped structure called the putamen, there are pathways that are the substantia nigra pathways, the black substance. They have a lot of black pigment because they have these melanin, which is the precursor to these neurotransmitters. But anyway, just basically look at this Brazil nut. It feeds this way, it feeds this way into the deeper pathways. This is the clutch of the brain. This couples the feelings, the intentionality, the actions. Okay? So when the clutch is slipping, then you have problems with movement. So again, as we alluded to earlier, the direct pathway is part of these dopaminergic or dopamine-containing cells. So you can't do stuff you want to do, like pick up a mahjong tile, for example. The indirect pathway is the inhibition, so loss of inhibition. All right, moving along. This is the same 4 slides I'll present at any talk if you go see a talk of mine or watch some of the later lectures. I finally figured out that it's only 4 slides people need to really get what's going on here. So I came up with this theory that it's really the shortening of the telomeres in the stem cells that causes the problems. In this case, arguably the differentiated neurons, but the point is simple. At the end of every chromosome are these tips called telomeres. Every time a cell divides, they shorten, and that results somehow in aging. They're like these shoelace aglets. So think of it as a firecracker fuse. It's burning, it's getting shorter. Here again, every cell has a nucleus, with the exception of skin and red blood cells. And in the nucleus is the coding, the library, and every one of these 46 chromosomes has a cap, which is the same in all people. Second slide is, what are stem cells? Stem cells are like queen bees. They are the master copies, and so she'll live years, but the drones around her are clones. They're clone drones, and they'll only live months. So again, keep in mind that there is an early progenitor cell, a common ancestor for neurons and the nurse cells. And so as this neurogenic or neurological stem cell copies, it gets errors. Just as copying a photocopy, you'll get streaks and blurs and speckles, rotation. So this poor queen bee is copying herself millions of times. There's going to be errors. And so the next copy is only going to be as good as the last. You can't liquid paper and retype this bad boy. It's damaged. So not only does the queen bee get damaged, but she gets older. So over time, her telomeres will shorten as well. The way that this is bad is that, and these 4 slides can go in any order, by the way. They're all 4 independent truths. As the telomeres get critically short, they're exposed, and the cell thinks it's damaged, so it tries to anneal them. It heals the chromosomes together in what's called an end-to-end fusion. So like these trapeze artists are now fused, let's say they're crazy glued here. As the cell divides and gives half the chromosomes to one daughter and half to the other, they're not going to separate the hands. They might separate the shoulders or the hips, the result being breaks in the chromosomes. So here, for example, the orange snapped off somewhere here, and maybe the maroon snapped off, the blue with the yellow. All these little crossings, there's triple here, there's no 20 here. There should be nice little pairs, but this is the process of DNA damage that occurs when telomeres get critically short. There's only one way that stem cells, well, the main way they protect themselves is through something called telomerase, which was the subject of the Nobel Prize in 2009. So the scientists who won the Nobel discovered that there is an enzyme complex which will write new telomere ends, sort of like an old stock ticker tape ticker, and it works not on all of them equally, but it would ignore this filled shot class. It would fill the shorter telomeres or the shorter shot classes, and that's how stem cells, despite copying so much more than other cells, can stay intact as they actively lengthen their telomeres. Okay, change subject. Apoptosis. Apoptosis is programmed cell death, and this is kind of a misnomer. Nobody really agrees on what they're talking about. It just means killing cells. And the major way that cells die is by killing themselves, like honorable samurai. So, you know, the cells copy, and when they copy, if there's an uncapping, there's a checkpoint system that if the DNA is damaged and the telomeres are not capped, this thing will need to be killed. It will kill itself. So in the case of end-to-end fusions, you get problems, again, we talked about separating in mitosis, and you get these tears and fragments. That will trigger apoptosis through a variety of mechanisms. Same here. When you get the telomeres uncapped, you can get apoptosis or just senescence, which is in some ways worse. You got cells that are just phoning it in. They're damaged. They're like, 64-year-old civil service employees. They're phoning it in, but they're not really doing the full job, and yet they're not willing to kill themselves. I'm not saying that's a cure to our current fiscal problem, but anyway, moving along, no politics. The mitochondria are made into bombs by p53. So the mitochondria are these power plants. They're the batteries of the cell. And so the way the cell will kill itself is usually by poking holes in its own battery acid bags, and that's how they die. The cellular junk that accumulates, such as the Lewy bodies, in Parkinson's in particular, is the aftermath, in my opinion. Not the party, it's the aftermath of the party. Another popular theory about Parkinson's is inflammation, and this will come up in a lot of diseases. People say, well, it's the inflammation. If you go to podcast 22 that we did 2 weeks ago on sports injuries, you'll see that inflammation is not the problem. Inflammation serves a purpose, the purpose being like a fire in the forest. It clears the debris, and the firefighters will come in and control the amount of clearing. There's an incredibly complex number of pathways, or there's an incredibly complex symphony of creative destruction that occurs in response to damage, but also in response probably to cell self-annihilation or apoptosis. So the key is telomerase activation within the neural stem cells, meaning neural stem cells keeping themselves young by activating their telomerase, keeping themselves protected. That's the trees. That's what regrows the damaged forest, just like we talked about in ligaments, muscles, bones, skin, in sports injuries. And also part of the repair is the microglial activity, which are cells I didn't explain, but they're the immune cells of the privileged brain space, which would be like worms, insects, bacteria, fungi, that are helping on a small level to help clear the debris and repair. All right, so before we do the literature search, let me just make a disclaimer. I am just an OBGYN by training. I'm not a research scientist, nor do I claim to be. So I basically do what anybody would do. I go onto PubMed, which is where all the articles are published, and I try and figure out what's going on. And that could be a good thing. It could be a bad thing. I mean, of course, when you enter a subject blind, you don't know what you don't know, so you make a lot of stupid assumptions. And, some of what I say may be laughable to a research scientist, but I liken it to kind of this caveman skit on Saturday Night Live where I think it was Phil Hartman would thicken his brow and he'd go in front of a jury and say, "Ladies and gentlemen of the jury, I'm just a caveman. Your world frightens and confuses me." And then he would launch into an argument. With that said, that disclaimer, he would sound like Clarence Darrow from Inherit the Wind, and it was kind of a funny juxtaposition there. So I don't know where I fall, somewhere between caveman and Nobel Prize winner, but my guess is that cells fail. So you see a party, a frat party, yeah, woohoo, they're raging on, they're having a good time, and these are the cells. Here is the culprit, right? The cup. The red cup is what causes these kids to poison themselves with alcohol or break windows or maybe start an unintended pregnancy. No, it's not the cup. But if you look at the literature in PubMed, there's 62,000 articles on Parkinson's. A great majority of them funded by billions of research dollars, and they're all looking at the cups. So this is the establishment. I think that's Cardinals in the Sistine Chapel. Again, you're not going to walk in here and say, golly, I think we should all worship, Shiva and Vishnu or whatever. It's not going to work. So when you look at the literature, they're looking at the red solo cups and the beers. They're saying, okay, we have Lewy bodies, we have pathways, we have chemical imbalance, we have genetic predilections and single point mutations. Again, their world frightens and confuses me. I don't claim to understand it, but I do think that what we can say about Parkinson's and other diseases is that the cells are bad, and that the cells go bad yeah, you could talk about oxidative stress or ultraviolet or viral cofactors, but cell information bad, cell function bad. That's basically my caveman interpretation there. Let's look at the literature briefly. The adage is, if you ask a stupid question, you get a stupid answer, right? And this is, unfortunately, about 85% of the literature in telomere biology is asking kind of a silly question. And what I mean by that is they look at leukocyte telomere length, which is what? Your blood drawn, your white blood cells, and they measure the telomeres. Yeah? Same with all the diseases. People will go, hey, telomere, that's cool. Let's look at heart disease. Well, let's look at cancer. Let's look at diabetes. And sometimes they find a shortening in the white blood cell telomeres. Sometimes they don't. But I liken it to checking the tire pressure to know if the carburetor is okay or the electrical timing. You check the tire pressure because it's easy to check. It's right there. Same with the blood. The whole blood's right there. But what you should be checking is the organ of interest or the carburetor or what have you, the timing. And so, lo and behold, if you check the white blood cell length in telomere patients, it's the same. So that was a stupid question. And that's unfortunately the state of most telomere literature. They don't measure the actual telomere of interest. Like if you're worried about esophageal cancer, then measure the esophageal telomeres. Don't measure the white blood cell telomeres, which vary tremendously, both within the person and within time and for different situations. Better question would be, what's the story with the cells in the substantia nigra? Okay, what's the telomere shortening there? Again, no evidence there. It's a dry hold. So, yeah, it could be unrelated, but are they looking at just the one functioning bulb in the many that are burnt out? You know, the crime scene investigation there doesn't really say how we got to that point. All they're saying is, yeah, the one light bulb functioning there still functions, so therefore, you know, light bulbs burning out has nothing to do with Parkinson's. Another thing that people look at in the literature of Parkinson's is myeloperoxidase, which is a pathway to destroy cells, commonly used by, attacking white blood cells. Multiple system atrophy is a disease that's similar to Parkinson's, and what they found with a mouse model is that if you inhibit the way of killing these cells, then the Parkinsonian symptoms improved. Well, that's really like saying, if you want to stop crime, then kill all the executioners. There's no pathway to kill or annihilate the damaged cells, but that doesn't mean that you did anything to stop the damage, which arguably could be from aging. I alluded to earlier that these mitochondria, which a lot of people blame for disease because they come into the crime scene, they see damaged mitochondria and oxidative species pouring out of here, like battery acid. And they say, wow, this must be the cause. But on the contrary, what we have is there's BAX, which the name is unimportant. There are these cool names, PUMA and NOXA. These are pathways that are used by the cell, and particularly P53, which we talk about at great length in podcast 2. P53 is the watchman. It's an amazing enzyme that sees if there's DNA damage. If there is, it says, whoa, we're not helping this cause, let's kill ourselves. And it pokes holes through the PUMA and NOXA in the mitochondria. The mitochondria leak battery acid, the cell dies. Okay? So apoptosis is a good thing. It's not a bad thing. It's how the cell kills itself. So everyone's looking at, again, the solo ups, and they're drawing wrong conclusions. So here, when you look for something, say, oh, you're getting warmer, you're getting cold. You go farther away from the hidden treasure. Well, here they're getting warmer. This is what I kind of infer for all the other podcasts, and when the literature is perhaps supporting this being just a theory paper, but this would be my bias, published in June of 2011, which is that P53 is the missing link between genetic and sporadic Parkinson's. Well, what is P53? P53 is just the way the cell recognizes it's damaged and wants to kill itself. So this paper is suggesting that P53 is the key factor, and that really is not a very profound statement. It's not really looking at the root cause. Again, as a caveman, I think the root cause is the cells are damaged. Here's apoptosis illustrated, P53 being at the very heart. And again, the problem is that the cells are getting old. They're recognizing that they're old. They're trying to kill themselves. So, just as though the oxidation is not the problem, the mitochondria is not the problem, the P53 is not the problem, the apoptosis is not the problem. You got to work backwards to the real problem. The problem is telomere shortening. Here, we're getting cold again. So, again, failed mitochondrial maintenance. So they look at these, and they go to the crime scene of Parkinson's disease, Huntington's disease, Alzheimer's disease, amyotrophic lateral sclerosis, which is Lou Gehrig's disease, and all the peripheral neuropathies, such as the stocking glove numbness in the twins in podcast 21 that improved while taking TA65. And they say, okay, mitochondria are not working well, and that these oxidative species are destroying the cells. This must be bad. Hence, the red solo cups must be making the frat guys get drunk. So that's unfortunately where science is going, is headed, and will continue to go, because the level of agency, the concept is wrong. The concept is that all this stuff, the police tape around the crime scene is what causes the crime. The red cups at the party are intimately, it's not really, doesn't make sense to me. I could be wrong. I'm just an OBGYN baby catcher, but doesn't make sense to me. Ellie was a 77-year-old woman whose son Bruce brought her in 8 months ago. Bruce had followed telomere biology for many years, very knowledgeable person, and he put her on it and took it himself, albeit at a lower dose. And after 4 months of taking TA65 4 times nightly, her processing speed in terms of moving the muscles, the hands, the fingers in a purposeful way increased by 116%. Psychomotor speed, as measured by this finger tapping test, we have this very advanced way of measuring your neurological function. It increased almost 3 times. Her lung capacity increased 28%, and this is only at 4 months, not even 8 months. We're going to measure her again soon. There was a minus 18% change in her ejection duration. Ejection duration being something that is bad. If your heart is working for a long time in the cycle to pump out blood, it means that it's not resting, and only during resting does the heart perfuse and other organs. And then the SEVR, which is a subendocardial viability ratio, she had a really low one, one of the lowest we've seen, which is an idea of how perfusable or how efficient the circulation is at perfusing the heart itself during the resting phase, and it shot up a tremendous amount from the danger zone into the safety zone after 4 months. Let me go ahead and take some questions here. Do you think the quick responses to TA65 is due to the lengthening of critically short telomeres or the activation of genes which provide benefit? It's multifactorial. TA65 will help the efficiency of the existing cells, I think, and the immune cells that clear damage, but I think the primary mechanism is one of apoptosis, meaning that the already damaged or marginal stem cells are killed, and they're killed and then replaced by younger, fresher stem cells. That's what I think is going on with most of these permanent changes. Because when a person goes off, like I went off for a great 10 months last year of TA65, I didn't gain any weight back. So what had happened was my fat stem cells in my omentum or my belly were killed for good. So that's the only way I can explain that, I think. It's cell death of damaged stem cells, which is, again, a good thing. What are the 3 takeaways I should know after today? Good question. Carrie, number 1, Parkinson's disease is the syndrome. It just describes where the damage is. It's not one real disease. It just means damage to those neurons. Number 2, the cures and the research focus on things that are not important, not as important. The tools of the cells, the pathways and everything. It's really the damage to the cells which are the cause of Parkinson's. So ways like stem cells of replacing those cells, or telomerase activation theoretically, although this is not an FDA proven statement, if the TA can kill the stem cells and they're replaced by healthier stem cells, then, or healthier neurons, then that would be a cure. And the third thing is that Ellie's Parkinson's got a lot better after starting TA65, and the other three things that she had taken for a great while. And Parkinson's being a progressive disease, she should have been getting worse, but rather she got better, which is unusual. Oh, question. Robert to everyone. Has Muhammad Ali or M. Michael J. Fox become aware of TA65? No, I don't believe so. I mean, we're still very early in the TA65 story. I wish I knew somebody that knew them. I think it's definitely worth a try. If nothing else, your mood, your sleep, and your skin improve right away. So that's encouraging. But again, something that needs to be taken over the long haul, I think, if it has any effect. And I would caution people to say everyone's results vary. There are complications which need to be managed by an experienced physician, and it's something that's legit. It's something that's definitely worth a try. And it's very rare that a person taking an adequate dose in the way that I recommend will say, I don't feel anything. They might say, I don't like what I feel. But if you come in and, again, objectively measure your heart, your lungs, your vision, skin, your cognition, as Eleanor did, you can come back to me in 4 months and say, you know what, Doc, it's not working. But then I can say, well, you did a pretty good job in faking your results because these are the objective measurements, and how do you explain this? And again, the beauty of this TA65 is that it's so gradual, it's so gentle, you can forget pills, you don't get withdrawal. People don't really think it's working. They just think that they've been endowed by the heavens with a better attitude and energy and lifestyle. So you really only appreciate it once you stop it and start it and stop it and restart it maybe 2 times. And you say, oh my God, yeah, this really is different. But it's not just different as a stimulant, because it's not a stimulant. It has no effect on your psyche at all. Are you doing something with your hair?
No.
Is that your hair, or is that
My hair?
a wig?
No.
Oh. Are you coloring it?
Oh, yeah, coloring.
Okay, yeah. It looks so full. I mean, when you get up in the morning.
I have a lot of hair.
Yeah.
Very thick hair.
What do you do in the morning when you get up?
Wash it. Not every morning.
Are you serious? So
Yeah.
how old are you?
I'll be 78 in March.
Now, have you noticed since starting the TA65 that your hair is more shampoo, that you need more shampoo, or is it thicker?
Well, my hair is thicker. I don't know whether it's since TA65, but maybe it is.
How about the skin? Is it tighter or smoother at all? Same?
Well, people say that I have very good skin.
Really? Now, you were telling me about a trip to Spain with your daughter.
Yeah, that was last year at this time.
And you just had to stop every 10 feet.
That was sciatica.
Was killing you. Yeah. How's the sciatica now?
It's not great.
Not great.
No.
Because when we talked a few months back, you had the fentanyl patch, but you had told the St. Joe surgeon you're not going through with it right now. Are you still not going through with the surgery?
No, I'm afraid of spine surgery. I really am afraid of it. 2 surgeons said the same thing, so I know that it's true.
Yeah.
This side is, I saw it in the x-rays anyway, this side is collapsed.
Okay.
And because of that, it's all crushed together.
Yeah.
And that's causing the sciatic pain.
Yeah.
So if they straightened it out and put bars in,
Yeah.
so I did bone density to see if the bones were strong enough to hold it.
Right.
I never did get an answer because I decided not to go through with the surgery.
I mean, you seem to be moving okay. Are you having sciatica at this moment?
Not while I'm sitting here, but I've had it today, I had it yesterday.
And the fentanyl, or do you take any painkillers right now?
I take a lot of painkillers and they don't work.
They don't work.
You know what works for the sciatica? Prednisone. And they don't want me on prednisone because you don't heal well with prednisone. It causes infection. I mean, there are a lot of bad side effects. But I go on prednisone if my asthma gets very bad. I must go on it. And that's when I discovered, while I was on it for the asthma, I suddenly discovered that my shoulder didn't hurt as much.
So you haven't done injections in the epidural space?
Yes, I have. I've had them all, and none of them worked. I had 3 epidurals before I went to Spain last year, and nothing. I couldn't walk a block.
So now tell me, about 2-3 weeks ago, the Mahjong gals were telling me that you were so bad for a couple of days that they had to drive you home. And you noticed that, and Bruce noticed that. So what happened there exactly?
Well, the only thing I can figure out, that happened after the fall, didn't it, Bruce?
Yeah.
That's the only thing I can figure out. I was stepping into the shower, turned on the water, and was stepping out the way I always do until it warms up, and somehow my body changed direction and propelled me to go head first into the shower, even though I was pulling back. I went head first into the shower, smashed my head, and skinned my whole arm off and cut my leg on the metal frame going through. I mean, it was a mess. There was blood everywhere.
Oh my God.
And that's the only thing I could figure out.
Well, that probably was the reason you fell, was the bad symptoms, not the cause.
I don't know. I don't know if the symptoms happened after that. That's what I'm saying.
Yeah. It's hard.
I went for a brain, CT of the
It
brain.
was fine, yeah. How many days did it last, the badness?
It was a
5
couple of
days?
weeks, wasn't it?
No, it was less than a week.
Like 4 or
Less
5
than a
days?
week that I couldn't drive?
Yeah, it was about 4 or 5 days.
That sounds right, yeah.
I thought it
so-
was longer than that because I couldn't, Mahjong, Judah says, what's wrong with you? I was flailing all over the place.
Yeah.
But anyway, yeah, I don't know which came first, whether the fall or the Parkinson's, but I was out in the parking lot and I was going to drive home,
Yeah.
and the manager came by and she said, I have never seen you look like this.
Right.
And I said, I can't drive. I couldn't move. I was really a mess.
Now,
So they drove me home.
do this for me?
Yeah, I do the testing every 3 months at the neurologist.
Yeah.
And each time he says, you're better than you were the last time.
How about the left side? This is what my wife does to me when I talk. She goes, I'm hearing this, Mom.
Now he has me do all this and touch
Let me
my
see.
nose.
Yeah, let me see. Touch your nose. You see the rigidity. So do this with your hand, and I'm going to test your rigidity here. Okay, look that way and do that with your hand. So you're not very rigid anymore, huh? Here, do it with this side. Tapping. Because we did those brain tests, and she was really good. I mean, her ability to do fast things was really good.
Mm-hmm.
Yeah, that's nice. Let's do a little walking, shall we? Stand up. How's your balance?
Well, it depends. My balance is good.
Hold out our hands like this, okay? Good. I don't see a lot of baseline tremor like when I first met you 8 months ago, so that's real good. And how about, okay, I'm going to stand behind you. Okay, I'm going to make you lose your balance a little bit. You ready?
Yeah, the doctor
Move
does this
forward.
all the time.
Okay, just look straight ahead. Stand up straight.
Mm-hmm.
That's pretty good. So you're a little better, huh? A little stronger. I like that.
Yeah.
Yeah.
Yeah, he says I'm better.
Or Parkinsonian type of gait in which the patient will have a posture in which he'll be stooped over, leaning forward, and then will have difficulty as far as initiating gait. When the gait is initiated, there are small steps. Oftentimes there's a tremor associated with this. And as the gait progresses, there may be a picking up of speed, or what's called a fenestrated gait. And then in turning, instead of having the normal turning, the patient will turn en bloc, which means they'll turn almost as a statue moving around.
Did you want to see me walk or anything?
Yeah, see you walk. Do the catwalk for me. Good? Yeah? I like it. Come on back.
And today I'm wearing heels, and I never wear heels.
All right, you're looking
I'm wearing
good.
boots with a small heel. And I'm fine today. My balance is just fine. 2 weeks ago, I could not do that. I was all over the place.
Amazing, yeah. You're ready to runway model.
That's it, runway model.
Looking good, Eleanor. So your posture's a little better too. Now, were you ever shuffling like this at all? Like
When my
looking down? Well, your back was hurting and then you're a little bit.
no, when my Parkinson's gets very bad, I can't move my feet.
Yeah. So move.
So I shuffle.
You shuffle, but you're not shuffling now. Like you take big
No, I
strides.
don't.
Well,
No,
look at you.
it's not bothering me.
Turn around. You don't have to. You don't have trouble turning either. That could be a problem too.
It is a problem if my pills aren't working.
So you're on the levodopa, right? So that's good.
And Mirapex and Selesmeline.
But you've been on that for
3.
years now, right?
Yeah, he upped one of them
Good.
from a half to a whole. That was the whole big deal.
Yeah. Well, Judith said that was the big difference, right?
That's what she said. Judith is the doctor suddenly. She said, what did your doctor do? I said, he upped the medicine. So she's saying now to everyone, well, that's why she's better.
Yeah. Have you noticed anything different in the last few months in terms of her movement, or not really?
She's up and down.
movements
Well, I mean, he upped the medication, but she was
for a while.
Okay, so you've noticed a difference then.
What do
Just-
you, what
with the
thing,
increased medication.
Yeah. So
what are you giving her?
it's a molecule from a Chinese herb that helps to repair the stem cells. But she said about 2-3 weeks ago she was really bad for a while there.
Very bad.
That was very bad.
That
We had to
happens.
drive her home.
Okay. Anyway, thanks for your attention, everybody. Have a great rest of the year. A Happy New Year. 2012 is going to be a big one. And again, everyone take care. Thank you. Bye-bye.
You keep saying you got something for me, something you call love, but confess you've been a messin' where you shouldn't have been a messin', and now someone else is getting all your best. These boots are made for walking, and that's just what they'll do. One of these days these boots are gonna walk all over you. Yeah.